Thank you to e-agricom for allowing me to share insights on antimicrobial resistance. I am a veterinarian, and I know this is a technical topic that has issues and challenges that affect everyone. As they say, health is wealth, and this talk hopefully increases awareness and promote understanding so we can all help in managing AMR. This webinar is a good way to address public perception on AMR. There may be extremes in understanding. Some of us may have no idea what AMR is, so this may be the first time you are hearing about it. Some of us may have already heard about AMR, and the take-home messages we often get is that antibiotics cannot be used in animals anymore. For this particular talk, allow me to tell you the technical definitions around AMR, and then we focus on one particular concern, antibiotics, and we will further focus on its use, especially in food animals. Let us start with some terminologies that you will most likely encounter around AMR discussions. First, AMR is antimicrobial resistance. This is the umbrella terminology, but the focus is really still on antibiotic resistance. However, we also encounter some confusion with antibiotic residues, so more about this later. Then there is AMU, or Antimicrobial Use, and AMS, or Antimicrobial Stewardship. Again, most studies and efforts would be focused on antibiotics. What we need to have a better appreciation of is the One Health impact of this AMR issue. This is not just about humans and animals having effective antibiotics to treat their diseases, but what is the effect as well on the environment or on the planet. The One Health approach is critical to managing AMR because resistant bacteria can spread, and antibiotics used to treat them are often shared between humans and animals. Then, there is also the concern of contamination of the environment. We can see that the issue of AMR, and specifically antibiotic use, will need to be a multi-sectoral approach. This is why talks like this are important to raise awareness for all stakeholders, whether you are the veterinarian consulting and prescribing for the farm raising food animals, or you are the farmer who play a big role in preventing infections, or the importer, distributor, or retailer of veterinary products like antibiotics, and as consumers, for all of us who need animal protein in our diets, we all have a role to play. If you try typing into your internet search browsers the words AMR and food safety, the first result is this website. And this can explain the importance of the issue and why we are focusing on food animals. Food plays an important role in the development and spread of antimicrobial resistance. Ingestion of AMR organisms or resistant organisms via food can, especially if they are pathogenic, result in human illness, and those illnesses might not respond to available antibiotics or other treatments. The presence of AMR microorganisms in our agricultural production systems and food chains is a potential route of exposure for everyone. This is why stakeholders in agriculture, and I assume that would be most of us in this webinar, are all enjoined to gain awareness and understanding of the issue. AMR means antimicrobial resistance. It might strike you as curious as to why the discussions are more on antibiotics. So it is always a good start with defining these two terminologies. Antimicrobials include antibiotics. In essence, antimicrobials are any of the substances which have a direct action on microorganisms, whether they are bacteria, viruses, fungi, protozoa, or parasites, and they are used to treat and prevent infections or infectious diseases in humans, animals, or plants. While antibiotics specifically are substances with direct action on bacteria. used to treat or prevent infections caused by them. Why is it important to understand the difference between these two terminologies? There has been an increasing trend in the use of the terms antibiotic and antimicrobial because there is increasing awareness and interest, especially to bacterial resistance to therapeutic antibiotics. I used to get confused and I used to say, they're just the same with each other, but they are not. So let's take, for example, this statement. It says, the problem of resistance, meaning bacterial resistance to a certain antibiotic, has been further exacerbated by the use of antimicrobials for viral respiratory infection. Considering that there are antivirals actually available and they are actually classified as antimicrobials, this has become a nonsensical statement. Now that we know the focus is on antibiotics, let us take a closer look. Antibiotics modernized medicine, from the discovery of penicillin, increasing the survival of human patients from bacterial infections, and to its eventual use in veterinary medicine for animals, particularly food animal production. The use of antibiotics in animals has ensured healthy animals are produced, and this means safe protein for the consumer and safeguards public health. Antibiotic use also helps veterinarians and farmers ensure the well-being of animals, treating them when they are sick. This also ensures animal welfare. This is why antibiotics are very critical to the animal industry. And this is also part of our efforts in sustainability. Sustainability means that we are meeting our own needs in terms of food, and hopefully without compromising the ability of future generations to also meet their own needs. It means we do not just take care of human and animal health now, we also take care of the environment. Even then, while he discovered antibiotics, Sir Alexander Fleming had a foresight already that the use of antibiotics might be abused. In 1945, he raised the alarm regarding antibiotic overuse when he warned that the public will demand antibiotics, and this will begin the era of abuses. The overuse of antibiotics clearly drives the evolution of resistance, as per his statement. This is why we see stringent controls in human medicine use, and we hope the same will follow with animal use. The 1950s to the 1960s were the golden age of antibiotic discovery. However, no new classes of antibiotics were developed since 1987. Since then, no new antibiotics were introduced with modifications only and adaptations to existing antibiotics. Just to give an example of this, macrolides, which were discovered in 1952, where you have antibiotic molecules like erythromycin used in humans, there were modifications like tylosin that are used in animals later in the 1990s, and the synthesis of telmicocin and the recent molecules like tulathromycin in the 2000s. Antibiotics have been developed and commercialized because they can be made by both natural and synthetic means, so they are already easy to make and have become a commodity. Most of us would be in the business that deals with antibiotic products. And the use of antibiotics have now come under scrutiny because we want to preserve their efficacy, both for human and veterinary medicine use. We only have limited antibiotics as tools, and we want them to still be available for addressing bacterial diseases. Let us focus on a key area of AMR, which is actually antibiotic resistance. We will use this infographic. We know what antibiotics are. Now we technically define antibiotic resistance. It is the resistance by the bacteria to an antibiotic, and this is what it was originally effective at treating infections caused by the bacteria. Resistance will not develop each time an antibiotic is used, but the more an antibiotic is used, the more chances there are for resistance to develop. To achieve our objective of managing AMR, all of us must understand that it must be antibiotic use in a responsible manner. You will also encounter discussions that suggest prudent or judicious use. The aim is the same, avoiding antibiotic resistance formation to preserve their efficacy. We also need to understand that antibiotic resistance is a natural defense of bacteria, and resistance can develop within a short period of time. It is not new. It was actually first identified in the 1940s for penicillin resistance, even before the antibiotic was released for widespread use. The first methicillin-resistant Staphylococcus aureus, you may have heard of it, or MRSA, was identified only 2 years after methicillin was introduced. But it is important to emphasize that this is not simply a result of antibiotic use commercially. It is a natural defense of bacteria. Antibiotic resistance is in fact ancient because microorganisms like bacteria have been present for millions of years, and antibiotics have been naturally produced by microorganisms. And this recent study have shown that bacterial DNA encoding resistance to beta-lactam, tetracycline, and glycopeptide antibiotics have been found in sediments from 30,000 years ago. It is as Darwin said, to survive, you must adapt to change, and for bacteria, that is developing resistance mechanisms. So, how does antibiotic resistance occur? Antibiotics kill most of harmful bacteria, but sometimes it is not able to kill all. Can you think of an instance where this can happen? Let me give you an example. My late grandmother would go to the doctor when she is sick and would sometimes get prescribed antibiotics for 7 days. After 3 days, she would feel well already. So she stops and keeps the antibiotics for any of her family to take if they also do not feel well in the future. In both cases, there is underdosing or the doses are not finished. And this is recognized in human medicine as a cause for resistance formation. The more sensitive bacteria are killed off early by the antibiotic, but because the full regimen was not finished, the more resistant bacteria may survive because they could already or have or developed a resistance mechanism. These resistant bacteria multiply and can pass on the resistance by genetic materials, and they can spread these to other bacteria that they can come in contact with. Let us backtrack a bit and talk about how does an antibiotic do its effect. An antibiotic will have specific targets for each target bacteria. This illustration shows us in red text the targets on each bacteria, and in black text the antibiotic classes that can have actions on these structures. Your doctors and veterinarians would know this. This is why we need to diagnose the causative bacteria in disease cases so we can recommend the proper antibiotics for it. This is researched and studied extensively before antibiotics are even registered and sold. So please use the product labels. So veterinarians use it, doctors use it, and actual field use and feedback on efficacy and other possible uses may be reported. That is why it is important to get updated. As an important note, there are certain bacteria that have intrinsic resistance. A specific example would be mycoplasmas. Mycoplasmas do not have cell walls. That is why penicillins and beta-lactams are not effective for them because they do not have the specific antibiotic target. Intrinsic resistance is important to understand because this is natural, and this is why product labels are very specific as to what the antibiotic is supposed to be used for. Since every living thing wants to survive, bacteria will protect themselves, including from antibiotics. by developing several mechanisms where they will acquire resistance. These are examples of the 4 ways, which are in red text, and in black text, the specific antibiotic classes that the bacteria may acquire resistance from. Now, it is important to know that this is what we term as acquired resistance. It is the resistance that develops when bacteria no longer respond to the antibiotic that was originally effective in treating it. As a result of this resistance, antibiotics and antimicrobial medicines become ineffective and infections become increasingly difficult or impossible to treat. This is the resistance we are concerned about. It also increases the importance of looking for other therapeutic options so that we can rotate the antibiotics or strengthening preventive actions so that we can avoid getting infections, like improving biosecurity. Now that we have established that we are looking at acquired resistance, it is important to understand how it is tested and what parameters are monitored to say that the resistance is already a big concern. First, we have the government-led AMR surveillance program. The bacteria that are monitored over time in a certain population and area. So samples are collected and bacterial isolation is done. These isolates are then exposed to different concentrations of antibiotics. Here in the illustration is the broth dilution test, and the results will read as MIC or minimum inhibitory concentration. These 4 intestinal or enteric and commensal bacteria, some of them actually do not give disease as they are normally seen in the gut. These ones are isolated because they are representative. The resistance patterns that they can give can represent for classes and for resistance genes. However, of concern to me as a swine veterinarian is a question of, is the drug I am prescribing for a herd or a flock still effective? This is when we submit samples to the diagnostic laboratory to determine the bacterial disease pathogen causing either the intestinal or respiratory infection. Some laboratories will then use the antibiotic sensitivity test, sometimes on agar plates, where the zone of inhibition, which is this clear space between that white antibiotic disc and the bacterial growth here, and the result will read either as SIRR or susceptible, intermediate, or resistant. Samples are gathered, bacteria is isolated, they are exposed to varying antibiotics, and they are checked whether they are resistant or still susceptible. How the results are interpreted can vary. In national surveillance programs, they do not all define resistance in the same way. They may be using the same parameter, but different interpretation standards. These country reports will use the epidemiological cutoff value because they're looking at populations, determining if the percentage of resistant bacterial isolates to a specific antibiotic is already a big concern. So, interpreting such reports must be done with caution and changes in these cutoffs noted. It is always ideal for each own country to generate their own AMR data because this data can be the basis for crafting of better policy options and not just collecting to say that there is surveillance. This would ensure the public that government is creating policies based on evidence. It is also important that all aspects are considered and the risk assessments are being done with stakeholder engagement. However, for a veterinarian worrying about whether his therapeutic decisions for the herd, it is the clinical breakpoint that are important. These are set standards to each antibiotic for each disease pathogen and are used to predict whether an antibiotic will be clinically effective against a particular bacterial isolate. They are based on a combination of MIC values, pharmacokinetic and pharmacodynamic values, and clinical outcome data. The result will guide the veterinarian if the antibiotics he is still using is effective in treating the bacterial disease. Hence, percentage susceptibility is important. If it is already low, then it may be time to rotate or switch to a different antibiotic class. Here are some added information, terminologies that you might encounter if you read research studies on AMR or antibiotic resistance. Terms like cross-resistance, co-selection, and co-resistance. And unfortunately, such resistance do not just come in ones or twos. Some bacterial isolates have been found to be either multi-drug resistant bacterial isolate or MDR. That means it has acquired resistance to at least one drug in 3 or more categories. An example would be TB in humans. There are also bacterial isolates that are extensively drug resistant, meaning they have resistance to at least one drug in all but 2 or fewer categories. So this means the bacterial isolates are susceptible to only 1 or 2 categories. So there are already limited options for treatment. And the one that we do not want to happen is the pan-drug resistant bacterial isolate, which is resistance to all agents in all categories. An example of this would be the PDRPA, that's Pseudomonas aeruginosa, and this is often seen in hospitalized patients. Some examples in veterinary medicine can include data on resistance to antibiotics used to treat swine respiratory disease, like Actinobacillus pluripneumoniae, Pasteurella multocida, and Streptococcus suis. The classification of a particular resistance phenotypes as MDR, XDR, or PDR could change over time as data for more veterinary-specific clinical breakpoints or antimicrobial classes and antibiotic drugs become available in the future. To summarize, now that we understand that there is intrinsic resistance, and we just have to refer to product labels to make sure that we understand which ones could be intrinsically resistant, and we now can focus on the more important acquired resistance, which is the concern, and we now know at how resistance can be passed on from bacteria to bacteria by genetic material, and this is where the current research is focused on new strain development at the genetic level. And there are many possibilities on how genes are transferred. They can be transferred by transformation, conjugation, or transduction. But there are caveats here. These are in vitro tests, meaning they are done in research laboratories, and the results should be interpreted with caution, as these may not be reflective of clinical or field efficacy of the antibiotic. So veterinarians' field experience, farmers giving feedback on treatments that have been applied and treating disease of the flock or herd, and the previous treatment records are very useful in this regard. We want antibiotics to be efficacious, to work every time we use them. So it is important to define how we use them. This is important to avoid misconceptions. There are 2 major uses, therapeutic use and growth promotion. Under therapeutic use, which is ideally recommended by a veterinarian with a prescription, first is the use for disease treatment. So when animals have clinical signs, they are treated with antibiotics. Second is for control or metaphylaxis. After diagnosis of clinical disease in part of the group and treating the clinically sick animals, antibiotics are also used to control the spread of the disease to animals in close contact, which are assumed to already have subclinical infection. This is important for food animals because most are reared in intensive production systems. Third is for prevention or prophylaxis, which is the use of antibiotics in animals or groups of animals before clinical signs of a disease. For us in swine production, where we take care of animals that have a long life cycle, there are stressful moments like the sows farrowing or giving birth, Are the piglets getting weaned from the sows or are being transferred, which may require transport? Veterinarians would institute a preventive dose, especially in light of historical health records, to anticipate the occurrence of the disease or infection. Then, there is the use of antibiotics for growth promotion, or AGP, which is to increase the rate of weight gain and/or the efficacy of feed utilization in animals by other than purely nutritional means. In this case, the use of antibiotics at subtherapeutic doses. This is now frowned upon, especially for medically important antibiotics. However, there is no clear guideline on animal use only antibiotics, which is actually not under scrutiny for AMR issues. Now, most antibiotic products have retained the treatment and control indications on product labels. Of particular interest now is the move of regulatory authorities to disallow the prophylactic or prevention use of antibiotics, especially in feed medication, because this is confused with AGP use. To clarify this confusion, we go back to our objective, which is to maintain antibiotic efficacy. We have seen that most regulatory actions currently are focused on in-feed antibiotic medication and arising from the confusion between the use as prevention dose or as antibiotic growth promotant, or AGP. The contention is that prevention use is the same as AGP use. We give here an example. Antibiotic A, when you read it on its label, you will see that prevention use is a therapeutic dose. An example given is 100 mg per kilogram body weight used for a defined duration. Example is 7 days. While previously, also in its label, you will see that its AGP use is a subtherapeutic dose, which is a very low 10 mg per kilogram body weight and often given continuously. If you remember how antibiotic resistance happens, in my example before, I pointed out that underdosing can allow a subpopulation of bacteria to survive, developing resistance. So, it is correct that we move away from using medically important antibiotics as AGPs, but we should not confuse it with prevention dose, which is often an essential therapeutic decision for us food animal veterinarians dealing with a big number of animals in intensive farming systems. The term medically important antibiotics refer to those that are important in human medicine practice. The preservation of the antibiotic efficacy is important for public health. There is an example for human use only, and that will be chloramphenicol. They should not be used in food animals. Majority of the antibiotics are shared between human and veterinary use. Ideally, these should be the antibiotics that should only be used for therapeutic needs in animals and only with veterinary oversight. You will see these listings in the World Health Organization list. We are also aware that there are still a few antibiotics that are only used in animals, and you will see them in the OIE list, or the World Organization for Animal Health. OIE is the French name for that. Now, both WHO and OIE are international bodies that give guidance. Why is categorization of antibiotic classes important? This is to further ensure proper antibiotic use to preserve its efficacy. Here we see the 6th edition of the WHO list of critically important antimicrobials, though this is still mainly a list of antibiotics. You will see 3 categories: critically important, highly important, and important antibiotics, and further there are highest and high priority antibiotics. You will also see more details in criterion and prioritizations. Note that the list defines antibiotic classes and generations, not the individual molecules. The WHO is constantly updating this list. referring to national reports on AMR surveillance and revising the antibiotic class categories as deemed needed. Some countries also develop their own categorizations and do not just rely on this WHO list. If we look at the suggestion of the WHO, and I emphasize the word suggestion because it is a guideline and not national policy, it is still up for countries to develop their own list. There is no prohibition on the use of antibiotics. Note that the orange bar, it is suggested that all listed antibiotics which are medically important, shared use between human and animal use, it is suggested to reduce usage and if possible to not use our prevention or as AGP, antibiotic growth promotion in food animals. While the one with the yellow bars, these are identified as highest priority or high priority, critically important antibiotics, and they are suggested that they are not used for control or treatment in food animals. So, what will a veterinarian use for therapy? As these are the bulk of available antibiotics commercially, what then can be a food animal veterinarian's use? Which also leads us to a question. If there is an antibiotic not on this list, so it is only animal use, so they can be used now for antibiotic growth promotion? So those are indeed things that we want to ask our regulators. Veterinarians get guidance from the OIE, and here we see the counterpart list. The classifications are veterinary critically important, highly important, and important antibiotics. So this list still does include mostly antibiotics. Same as the WHO, it lists by antibiotic classes and generations, not individual molecules. When I attend webinars with the WHO, the OIE, and the FAO, the tripartite UN organizations helping with AMR, we often would get comments that the WHO list is unfair to veterinarians, and I could not agree more. There is a need to harmonize the WHO and OIE lists because veterinarians still need antibiotics in their toolbox for treating animal diseases. So, if we have allowed antibiotics, there are also banned ones. Here I have put 3 similar lists. First, the FAO, or the Food and Agriculture Organization's Codex Alimentarius. It shows a list of veterinary drugs where risk management is recommended, and so no MRL or maximum residue limit is set. Simply put, no residue is allowed in the meat, so it is not allowed to be used in food animals. Second, for us in the Philippines, we have the PNS, or the Philippine National Standards, as issued by the BAFS under DA. And finally, from BAEDA, a list of banned drugs and antimicrobials. Highlighted in blue text would be the antibiotics banned for food animal use. All other antibiotics as per PH policy, as long as registered in both FDA and BAE, are allowed for use. Since we touched on residues, let me add information on the difference with resistance. There are misconceptions that antibiotic residues can cause antibiotic resistance. So far, there is still no data to connect the two, and we should clarify instead of spreading fear as it diminishes the food safety assurance of animal protein producers. A residue is a chemical hazard. It is the antibiotic itself or its metabolite in the meat of animals. It can be determined with an ELISA test or an LCMS test. We already know that resistance is microbial. It is either the resistant bacteria or genes, and testing it is by bacterial isolation and antibiotic sensitivity tests. To intervene and avoid antibiotic residues, it is important to follow product label indications, especially withdrawal periods, to lessen the risk of having residues in meat. On the other hand, antibiotic resistance intervention or avoidance may be done with stewardship programs. Veterinarians play a major role in mitigating AMR by adopting AMS, like responsible use of antibiotics. Although these two issues are unrelated, they are both important, and understanding the technical difference and the approach to address them are key. It has been proposed that the antimicrobial or antibiotic stewardship for practicing veterinarians can be the statement on responsible use of antibiotics. As a veterinarian, we get the right diagnosis, use the right drug in the right amount or dose, the right route or the escalation for the right period or duration, and through a prescription. In the Philippines, that is the VDO or the Veterinary Drug Order with a VCPR or the agreement with the farmer. In a previous talk, the PCSP made an informal survey, and these are some of the comments related to the antibiotics listed here. Veterinarians emphasize the need to communicate more effectively so we can gain stakeholder support. And I hope that we can achieve that today. Another veterinarian advised to use registered products only, and a pharmacist raised the concern on seeing many unregistered antibiotic combinations on the market. All of us have a role to play, and all of us can adopt an antibiotic stewardship program. This is to meet the objective of preserving the efficacy of antibiotics. We have already mentioned the actions that food animal veterinarians can do, and all of these can be summed up in having veterinary oversight in the use of antibiotics. This would also be reflected in the farms that they advise and consult for. These are the suggested actions. For our counterparts in the government, doing the regulatory control, these are suggested actions. The pharmaceutical industry play a big role, and these are the suggested stewardship actions. And those in the allied industry doing selling, dispensing of antibiotics, these are also suggested actions. We all need to understand how antibiotics can impact the products like meat, eggs, and milk as these move downstream to the distribution chain. How will it impact customer and consumer perception? How will it impact public trust? And what about our social licenses in making sure that we move our products? How can we ensure that all of this is done with the aim of protecting the efficacy of antibiotics? When you look at discussions around AMR and AMU, the assumption always is that it is imprudent use of antimicrobials in food production, including livestock, aquaculture, and crop production, that is prevalent. And the use of such, like antibiotics, including sanitizers and biocides, through the food chain may further contribute to AMR. Part of managing AMR is increasing awareness. One of them also is supporting government initiatives in AMR surveillance. As an industry, we need to show that we are being prudent or responsible in the use of antibiotics, and that would include showing data that we can monitor how and by how much to use antibiotics. Of particular interest is how we sell veterinary drugs and antibiotics in the Philippines. We have importers who sell down the line to manufacturers or distributors and on to selling and retail channels. How do we capture use data? Where do we put controls? How can we show we are responsible in use and not doing indiscriminate use? Efforts to quantify antibiotic use are already underway. This is the data from 2016 from OIE's 4th annual report on AMR, and we see that Asia has the highest use of antibiotics. This is the 2017 data as reported in the 2021 OIE's 5th annual report, and in here there were 10 more countries, and all, except the Americas, showed lower quantities of antimicrobials or antibiotics reported. Asia remains to have the highest quantity reported. Some context is needed. These reports covers all species, all molecules in all regions, and all their own flocks herds temporally. So we need a scaling factor so this data can be comparable to each other. To be able to compare data between countries and regions, we need to use a scaling factor. That's why the OIE contextualized a denominator, the animal biomass, and it is now used in many in AMU estimation methodology. This is true for both human antibiotic consumption data as well, and now in animal, especially for food-producing antibiotic quantities. The OIE provides calculation tables and biomass factors that can be used, so countries need to get a good grasp of our animal population numbers. What are concrete actions that we can do to help manage AMR? We know that a growing world population and the resulting increase in demand for food is putting added pressure on our food supply chains and systems. There are many systems and many conditions in our farms that can favor the selection of antimicrobial-resistant microorganisms, and they can cause human illness or indirectly damage food production. One of the things that farmers can do, that veterinarians can help with, is establish good hygiene practices in agriculture. This is fundamental in achieving food safety and is key in addressing AMR. Biosecurity and other prevention practices are something that we have the power to do for our farms. This brings me to the end of my talk, and I would like to thank all of you for your interest. I hope you now have concrete examples of how to manage AMR, and you have understood the issues around the use of antibiotics, especially in food animals. Before I end, I would like to thank Dr. Agnes Agunos and Dr. Caroline Benigno for adding their insights to help align the messages. To all who have gained awareness, thank you for the interest.